Yeah, is he ready?
That
Okay.
sure is a big disappointment compared to the last one.
Well, we doubled the audience, right? Good. I will talk about impacts of rising CO2 on harmful cyanobacteria. I'll first take you here to this area. This is actually not so far from here. This is 150 miles west of Cambridge. This is along the Dutch coast, the southwestern part of the Netherlands, a province called Zeeland. And as you can see, it used to be an area with the influence of the North Sea over here. And then all kinds of estuaries here, and the River Rhine and the River Maas were coming in here. The Scheldt is coming from below. So it's a real estuarine area. But
they've reclaimed a lot of that land.
yeah, but the major disaster first happened in 1953. There was a huge flooding, both here in the Netherlands and also in the UK, which flooded most of these islands that you saw. It flooded also large parts of Norfolk and Suffolk here along the coast. It was the largest flooding disaster we have experienced in this part of Europe. And for that reason, the Dutch decided to build a system of dikes, dams and sluices known as the Delta Works. So you can see all the new construction works that were built over here. And it radically changed the system, of course, and in particular, some of the systems that used to have salt water actually were turned into freshwater lakes. And one of them is this lake over here. This used to be an estuarine area, but it's now an enclosed lake, an enclosed freshwater lake. It's known as Lake Volkrak. And what happened in this lake, so we changed it from an area with tidal motion to a stagnant freshwater basin. And within a few years after that, as soon as the water turned fresher, we got a huge development, massive developments of cyanobacterial blooms. This is from an airplane. You can see the airplane over here. And this is a coverage, a dense coverage by the cyanobacterium called microcystis, because they like fresh, stagnant conditions with very nutrient-rich waters. Now, what this organism does, microcystis, is it produces a family of toxins known as microcystines. This is the molecular structure of microcystine. It consists of 7 amino acids. And these microcystines, they are toxic. They act on the liver. They poison and can actually kill your liver. And there's also wide diversity. About 70 different variants of microcystine have nowadays been described, each with a different combination of amino acids. So these are toxic compounds for us, for mammals, for birds, and so on. And what happens in these very dense blooms is that these microcystis cells, they may float upwards towards the surface. Many of these microcystis cells have so-called gas vesicles that provide the cells with buoyancy. They can float upwards and they form very dense scum layers. Sorry for the Dutch here. Drijflaag, but it means a scum layer at the surface. So what we have in this case, in this lake, is this is the concentration in the open water of microcystine, the toxin that's produced, and it's in the range. And it's also here indicated by these purple symbols here. It's in the range from 20 to 100 micrograms per liter, and the guideline value from the World Health Organization is 20 micrograms per liter. Above that, you shouldn't swim there, and above 100 micrograms per liter. You shouldn't use it to make drinking water. What you find in these surface scums is that the concentrations go up, and then we have to look at the right-hand side to values of 4,000 to 5,000 micrograms per liter. Right, so that's 200 times the guideline value of the World Health Organization. These are very toxic scums. And what happens in several years in Lake Volkrak is that we had mass mortalities of birds, and basically all the birds that just wade through the water, like ducks and swans and gulls, and also here this spoonbill, a red list species. So that's an example of what happened in our country in Lake Volkrak. But there are also other problem areas in the world. One is in Lake Taihu. It's the 3rd largest lake in China. It has a surface area of more than 2,000 square kilometers. And also this entire surface area in some years is completely covered by microcystis blooms. In 2007, the problems were even that dramatic that it had to be decided that the lake could no longer be used for drinking water, and as a result, 5 million people were without tap water for more than 10 days, which caused an enormous rush on the shops. They also run out of bottled water, so you can imagine what kind of problems this can create for large cities. Other famous problem areas with toxic cyanobacteria are the Baltic Sea. Here you see pictures of the area between Sweden and the Baltic states. And this is the island of Scotland. Here you see a real-world photograph of it. These are all toxic cyanobacterial blooms that expand over hundreds and hundreds of kilometers. This is an aerial photograph. Here in white you see the clouds, and underneath here you see the blooms of the cyanobacteria. And also in the United States we have similar problems. For instance, last year there were major problems with Lake Erie, and the city of Toledo was a couple of days without drinking water because there was a too dense Mycocystis bloom in that area. Okay, if you summarize it, the problems caused by toxic cyanobacteria is that many of these lakes have to be closed for recreation. You're not allowed to swim there if the concentrations of cyanobacteria are too high. It's dangerous for dogs and cattle. If they drink from it, you might have some kills there. It's dangerous for birds. You cannot use the water for drinking water. You cannot use it for shell fisheries. And you cannot use it as irrigation water in agriculture because the toxins will stay on the crops.
You haven't mentioned fish.
I haven't mentioned fish, no, because actually it turns out that quite a number of fish species have no problems with the toxins. They can have problems with oxygen depletion, but they're not as troubled by the toxins as birds and mammals.
What is the purpose for the blue algae to produce this? Do they protect?
That's a major debate, and it's a major question. One hypothesis is that it deters grazing from zooplankton.
What?
That it deters grazing by zooplankton species.
By zooplankton.
Yeah, but the thing there is that actually many zooplankton species are not harmed that much by the mycocystin. And the other thing is they also looked at divergence of the mycocystin genes and estimated when it evolved, and probably evolved before the protist developed. And that's also kind of problematic. So there's a huge debate going on. What is the function? Did it have or does it have another function? And it simply turns out to be toxic for us. Yeah. So here's a case study from a lake in Amsterdam. That's Lake Nieuwmeer. That's a recreational lake in our city.
I'm serious, what if it's antiviral?
Yeah, there are certain viruses known for mycocystis,
Yeah.
and what can happen is that they just clear a lake from mycocystis in a couple of days. Yeah.
So things like that are really susceptible to that. So what if you have an antiviral? Are you guys ever checked?
Yeah, people are trying all kinds of different measures against mycocystis.
No, I mean your toxins. Have you ever checked to see if the toxins are antiviral?
If toxins are antiviral, I'm not aware of that. No, I don't know whether people have ever checked this.
Because
It's interesting. Yeah.
because there's like all of that. That's always the game that you're playing whenever you go
Okay.
into like monoculture-ish sort of stuff. Okay, that's fine.
Good question. Okay, we're back in Lake Nieuwmeer, recreational lake in Amsterdam. The problem also here is that we have this mycocystis, and as I told you, mycocystis is a buoyant cyanobacterium. It produces gas vesicles. What you see here in this picture is a colony of mycocystis cells. So each round circle here is a cell, and you see also within this cell some structures. And actually, the light areas are the gas vesicles of mycocystis. Now, if you have these large colonies filled with cells with gas vesicles, they can float upwards, and they can form these very dense surface clumps.
Sorry, what's the white stuff?
This other white, this very broad white stuff is extracellular polysaccharide that keeps the cells together, and the white is within within the cells are the gas vesicles, so the different scattering within the cells. So they float upwards as colonies, rather loose colonies, forming these dense surface scums. And as a result of that, Lake Nieuwmeer had to be closed for recreation almost every summer, whereas it was a recreational area within Amsterdam. So that was a major problem. And the water management asked us, can you find some kind of solution to get rid of these microcystis blooms? And we thought, well, one thing that you might try is to mix the entire lake. Mix the entire lake by bubbling it with air. And the idea is that if you mix it faster than the flotation rate of the microcystis cells, then they are no longer able to form surface scums. They will be homogeneously mixed through the water column, and then they lose their competitive advantage. Because if they can form these surface scums, then they themselves are in the light, and they shade all their other competitors. They shade the green algae, the diatoms, and other phytoplankton species. So they have a competitive advantage if they can form dense blooms over other species. If you can take that advantage away, maybe you get rid of the microcystis bloom. So the city said, well, let's try whether that idea works. So they built a whole installation in the lake, consisting of 7 different pipes, 2 compressor installations, and it's compressing air through these pipelines. These pipelines are perforated, and we bubble the entire lake. You can see it from the air if you fly into Amsterdam. Then if you go to the airport of Schiphol and you look out of your window, you see a lake with all kinds of streamlines from the bubbling of these 7 pipes. We combined it with the development of a model. It's a one-dimensional model that looked at the competition between different phytoplankton species. And for each species we had one of these equations. And it gives you the population dynamics of a species. So this is the change in the number of species I as a function of its production, its losses, its vertical velocity by buoyancy or sinking, and its vertical mixing by turbulence. So it's a 1D model. The production, we assumed that nutrients are saturating because it's an extremely nutrient-rich lake. But the production rate therefore mainly depends on the light availability. And these are the specific growth rates of the different species as function of light intensity. And what you can see is these buoyant cyanobacteria, this microcystis, doesn't have a very high maximum growth rate. Actually, it's much lower than the maximum growth rate of, for instance, diatoms and green algae. And the idea there is that this cyanobacterium invests a lot of its energy in its gas vesicles, and as a result of that, it has less energy, less material to invest in growth. So that's the production term. The loss term was just a species-specific constant. The buoyancy or sinking, well, the cyanobacteria are buoyant. They have a certain flotation velocity upwards. And for instance, the diatoms, they're sinking, so they have a certain velocity directed downwards. Then we have the turbulent mixing of the water column, and what it does, it redistributes the populations over the vertical. And this turbulent diffusion coefficient is a property of the water column itself, so it's the same for all this value of D is the same for all the different species. But what we do by the bubbling of the entire lake, by the mixing of the entire lake, is that we vary the value of this turbulent diffusion coefficient. Right? We increase it by bubbling the lake. So here are the model predictions. What we did is we run this model many times for different values of the depth of the lake and for different values of the turbulent diffusion coefficient, this D. And each time for each simulation we wrote down like what the outcome is, who will win and who will lose the competition, and then you obtain a plot like this, like in this entire area for all these combinations of lake depth and turbulent diffusion coefficient. We find that the buoyant cyanobacteria win. In this area over here we find that the diatoms win. In this area we find, in this hatched area, we find coexistence of the cyanobacteria and the diatoms, but it's only a very narrow area. And this area here, none of the species could grow. So the idea is that the lake used to be in this state, where the cyanobacteria won the competition with low turbulence, and that by bubbling we changed the system towards a state like this, much higher turbulent mixing, so we shifted from the buoyant cyanobacteria to the sinking diatoms. So we could also advise like what critical level of mixing was needed to shift the system from one state to another state.
Yeah.
Yeah?
So as water column depth increases, cyanobacteria will win. How to explain this?
Yeah, let me explain it in this way. If you have a very small cup of coffee, you only need a little bit of turbulence to mix the diatoms through the entire water column. If you have a very deep lake, you need much more intense mixing to get the diatoms mixed throughout the lake. So that's why you have this line.
I mean, are diatoms really sinking at an appreciable rate?
The ones in our lakes, yes, because we measured their sinking speeds. Yeah. It depends a bit on the species, and it depends also on whether you're in freshwater or marine systems. But especially in freshwaters, most of the diatom species are sinking because they have the silica shell and it has a higher density than water. So they tend to sink. In marine systems, you have some diatoms with very large vacuoles that they fill with fresh water, and then that gives them some counterbuoyancy. So in marine systems, it's a bit different. But in freshwaters, most of the diatoms are sinking.
Because in deep water, it's obviously, they have to, they can't just sink.
No, but if you look at what happens if they tend to sink, but there's also the redistribution of the population by turbulence. So it's not that the entire population just ends up at the lake sediment, but it's also they slowly sink and then they're remixed again, they slowly sink, and then they're redistributed again by the mixing. Yeah. So if the mixing is sufficiently high, you keep them suspended, and you also avoid surface layers of the buoyant cyanobacteria, and then the diatoms win over the cyanobacteria. So that's what the water management did. They started to mix the lake. They switched the system from this level of turbulence to this level of turbulence by artificial mixing. And this is a typical year when the mixing was not yet there. And we always had cyanobacterial blooms in summer. And this is how the lake looks nowadays. So this was since the mid-1990s, the lake is mixed every summer period. And we are almost rid of the cyanobacteria. Sometimes you see a small trace of cyanobacteria here, but that's about all. Good, you would say, well, you found a solution, problem solved. But the thing is, mixing an entire lake is very, very expensive. And you have to do it every year, because if you don't mix, you get the cyanobacteria back in summer.
Have they tried that?
So they asked us, yes, they've tried that. Yeah. They asked us, well, can you think of a way to reduce the cost? And we thought, well, perhaps we can halve the cost if we switch the mixing only on half of the time. So we do an on-off mixing regime. So we negotiated a bit with the water management what the ideal on-off mixing regime would be. And in the end, we came up with a mixing regime of 2 weeks on and 2 weeks off. And the reason is they can't mix it on and off every day, because that also costs a lot of energy. Plus the persons who have to go to the lake to see whether the mixing installation works properly. So they said, well, you will only save us money if we can have like relatively long periods, like 2 weeks on and 2 weeks off regime. But they were prepared to do this, and for us it was very exciting, because we thought, well, this gives us a new scientific challenge, because we can see what happens with the plankton community. And we also had some models developed where we coupled plankton competition with changes with the hydrodynamics of a lake. So we coupled these models and investigated a coupled model of plankton dynamics, hydrodynamics, and weather input, meteorology. So that's what we decided to do. But when we made this decision to have this on-off regime, we didn't know that summer it would be an extremely hot summer. So this was. We would do this experiment in the summer of 2003, and it was going to be the hottest summer in the past 500 years. Here you see the summer temperature measured at the Royal Meteorological Institute in the Netherlands for 2003. You see the increase in temperature in summer, but this is 2003. Now what happens when you have an increase in temperature, what happens to lake waters? Well, first is that at high temperatures you get a much stronger stratification. As most of you will know, lakes can be stratified with a warm top layer, so-called epilimnion, on top of a colder bottom layer. And the larger the temperature difference, the larger the density difference between these two layers, and the more stable the stratification is. And this is of advantage for buoyant cyanobacteria, because the calmer the water is and the strong stratification calms down the water, the calmer the water is, the more easily they can float upwards. The second thing is that temperature affects the viscosity, and what happens is that the viscosity affects the vertical velocity of microorganisms. That's determined by Stokes' law, and according to Stokes' law, the vertical velocity is inversely proportional to the viscosity, and the viscosity decreases with temperature. What that means is that higher temperatures, the sinking species, the diatoms, sink faster, but the buoyant species, the cyanobacteria, float upwards faster. So again, an advantage for buoyant cyanobacteria in comparison to their competitors, the diatoms. The third is we also looked at the temperature dependence of the growth rates of our organisms in Lake Nieuwmeer. And it turned out that the green algae and diatoms had their optimum growth, the maximum growth, at a temperature of about 20 degrees centigrade, but Microcystis prefers warmer waters. It has its maximum at about 28 degrees centigrade. So that's, again, if you have a summer heat wave, it's again advantageous for Microcystis in comparison to the diatoms and green algae. So here are some measurements during our 2003 experiment. We had a temperature logger installed in the lake, and we had our model predictions, and you see here the measurements in the simulation of temperature at 4 meters depth. And you see the effect of the switching on and off of the mixing regime, and you see also that the lake water temperature increased tremendously up to mid-August. And here you see, first you see again in the top graph, you see these effects of temperature, mixing off, mixing on, mixing off, mixing on. Highest temperatures here in mid-August. This is the development of the phytoplankton community. And what we got is, first when the mixing was off here, we got a little bit development already of the cyanobacteria. Then the cyanobacterial numbers were brought down again by turning on the mixing. But when the mixing was switched off again, we got a huge peak of cyanobacteria. And actually our experiment failed. We could not suppress a cyanobacterial bloom. At this point, the water management decided, okay, we stop the experiment, we continue mixing of the lake, because else we have to close the lake for recreation. So mixing was switched on again, and now again, every summer again, the mixing is on from the period till April, till October, just the entire summer period. The lake is completely mixed. For us, the question was like, so what happened here? We had this on-off mixing regime, and we also had an exceptional year with a very, very extreme summer heat wave. So we decided to make a model comparison, because we had built our models of phytoplankton competition and hydrodynamics and weather input, and then run the model for 3 contrasting weather conditions. We took the weather of a very cold year, cold summer 1956, an average summer and a very hot summer. And based on that, we can conclude that this year, 2003, was really a disaster for us, because basically that allowed the strong increase of the cyanobacteria. So key conclusion, summer heat waves brought blooms of harmful cyanobacteria. Now that's the effect of temperature. And since then, many other people have looked at effects of temperature on cyanobacterial blooms. But if you think about global change, there's also another problem, and it's the direct effect of CO2. Because phytoplankton species and cyanobacteria, they feed on CO2. So what about the effects of rising CO2 on cyanobacterial blooms? Well, then we first have to go into a bit of carbon chemistry. So here we have CO2, and if CO2 dissolves in water, we get bicarbonate here, HCO3- and carbonate. Also what you see is that there's a production of protons, H+, so you get a lower pH, that's known as ocean acidification. I think most of you are familiar with that phenomenon, that the oceans get more acidic because of the dissolution of CO2. Now for phytoplankton, what's this? Green. Okay. What phytoplankton consumes CO2, and most phytoplankton species can also consume bicarbonate. They cannot consume carbonate. So what happens if you have consumption of CO2 and consumption of bicarbonate? Then the reactions will go from this side upwards to here, and it's consuming protons. So during dense phytoplankton blooms, the pH will go up. So remember again our Lake Volkrijk, the lake where we have these dense cyanobacterial blooms in the Netherlands. And these are data from Lake Volkrijk during 2 years, the year 2000 and the year 2001. Here you see the 2 summers with the cyanobacterial bloom, very dense cyanobacterial bloom in this summer and also in this summer. And here you see the concentrations of CO2. The dashed line is the CO2 concentration that you would expect if there would be an equilibrium with the partial pressure of CO2 in the atmosphere. And the red line is the actually measured CO2 concentration. So what you can see is that there is a so-called supersaturation of CO2 in winter. Do you know why that is?
What?
Why you have supersaturation of CO2 in winter?
The reaction is not at equilibrium.
Yeah.
So
So it's because many of these lakes receive a lot of organic matter, and bacteria start to mineralize the organic matter, so you have a lot of CO2 production in winter. So you have supersaturation in winter of many freshwater lakes, but you have undersaturation in summer, and that's because all the phytoplankton takes up the CO2. So here the actual CO2 concentration is much lower than what you would expect based on equilibrium with the atmosphere, because the phytoplankton bloom is consuming all the CO2. And as a result of that, what you observe is a strong increase in pH. In this case, the pH goes up to about 9.5 in summer when the CO2 is strongly depleted. Now the question is, what will happen in such systems when you have a higher atmospheric CO2 pressure? So to investigate this, we developed a model where phytoplankton growth is a function of CO2 availability, bicarbonate, light conditions. What happens, we developed the model in 3 different steps. First, you have those resources. The carbon is taken up by the phytoplankton cells. Then it's stored, and depending on the amount of storage determines the growth rates of the phytoplankton population. So this is basically the idea. This is in terms of mathematical equations. So we have an equation for the biomass. We have an equation for the cellular carbon storage in the biomass, consisting of uptake of carbon, respiration, and dilution of the carbon in the cells by growth. We have equation for the carbon chemistry, for the dissolved inorganic carbon, and for the alkalinity. And from this we can also calculate the CO2, bicarbonate, carbonate, and pH in the system. And we first decided to test this model in laboratory experiments. So here are our laboratory chemostats. We used microcystis strains taken from Lake Volkrijk. And here are two of these experiments are shown. This is one experiment at a low level of pCO2, 200 ppm of CO2 in this experiment. The bottom experiment has a high CO2 level, 1200 ppm of CO2. And what you see first is that when these chemostats are occulated with a little bit of microcystis, the population density increases. As a result, the light penetration through the chemostats declines because they become greener and greener, they become denser and denser. Light penetration decreases. You see changes in the distribution of the inorganic carbon. So the CO2 is taken up preferentially, so that means the CO2 is strongly depleted. But for instance, because of the CO2 depletion, you get an increase in pH, in this case up to about a value of about 10, which shifts the carbon towards bicarbonate and carbonate. So you see a strong increase in the carbonate concentration. So all these kind of shifts are taking place in one single chemostat. And the symbols here are the data, and the lines are model predictions based on the model that I've just showed. So below we see the experiment at 1200 ppm. What you can see in comparison is that the population density is much higher than here. What you can see is that the CO2 is depleted much less at high pCO2 levels, at low pCO2 levels. What you can also see is that pH still rises because you still have consumption of CO2. So the pH should rise, but here it rises only to about a value of about 8.5, as here it was increasing to a value of about 10. So it rises to a much lesser extent. Now, what you also observe here is that the experiment runs to a steady state. So this is a chemostat experiment, and in the end, after about 25, 30 days, the system settles at a steady state. And what we can do is we can run many more of these experiments and then look at the steady state properties. That's what we did in these graphs. So here each dot is a single experiment performed at different pCO2 levels. And here, for instance, you see the steady state population density as function of pCO2. So what you see first is an increase in population density with rising CO2. But then at a certain point you can add more and more CO2, but the system is carbon saturated, and you don't see any further increase in the population because it gets limited by something else. In our case, it became limited by the light energy. So we supplied a lot of nutrients. It was never nitrogen or phosphorus limited, but it became limited by light. Simply it couldn't produce more biomass from the light energy that we provided. What you also see is a decrease in the light penetration through the cultures. If they're more dense, then you get less light that penetrates through. You see an increase in the CO2 concentration, a decrease in pH at higher CO2 levels, an increase in bicarbonate, and an increase in DHC. So these are the expectations of what will happen during dense blooms with increasing pCO2 levels, at least based on our models and our chemostat experiment. So the next step was to extrapolate or find some way to extrapolate these laboratory findings on the chemostat towards a real lake system. And basically what we did is we took our model, but we looked like what things are now really different between a lake and a chemostat. Well, some things are very obvious. For instance, the chemostat is only very small, whereas a real lake might be 5 meters or more deep. The CO2 exchange rates are very different between our chemostat and a real lake. The residence time is different. There might be additional grazing. There might be burial of phytoplankton, which we also took into account into our model. So we added various components that we thought this is really relevant for a lake, and then used the parameters that we had estimated, measured for our microcystis populations, and run the model for a lake scenario. We have many results, but I only show you the summary graphs here. So this is for a lake scenario. And here we run simulations at different atmospheric pCO2 levels, and in the top graph, different alkalinities of the lake. Because you can have low alkaline lakes, and you can also have very high alkaline soda lakes. And then we looked at what is the level of carbon limitation in the lake. What you observe is already in line with what you expect. So lakes with a very high alkalinity, they have a lot of bicarbonate. And cyanobacteria can also use bicarbonate for photosynthesis, so they will not be carbon limited in very alkaline lakes. The level of carbon limitation is very low. But in low alkaline waters, you have much more carbon limitation. And with an increase in atmospheric pCO2, this carbon limitation will be alleviated. Right? Because you're adding more and more CO2, so they will have less problems with the carbon limitation. And this is also what this pattern then reflects what will happen with the population densities of the phytoplankton blooms. So in very alkaline lakes, you do not see much effect of the rise in pCO2 concentration because they already have very dense cyanobacterial blooms because there's no limitation by carbon in those lakes. But in the low alkaline waters, we expect strong increases in phytoplankton blooms because with the addition of CO2, you alleviate these blooms from carbon limitation, and that means you get more and more potential for bloom development. Okay, so our expectation is a strong increase in phytoplankton blooms at high pCO2 levels. But now one of the questions is, of course, like what will happen with the traits of these organisms? Because it's not only that they might increase in biomass, but they might also adapt to the higher CO2 levels. Now, to investigate this, we have to look a bit deeper into the way these organisms use CO2. And many cyanobacteria, they have a so-called carbon concentrating mechanism. It's depicted here. So here we have a cell of a cyanobacterium, and they have all kinds of transporters for CO2 and for bicarbonate. This brings the bicarbonate into the cell. The bicarbonate is transported towards a so-called carboxysome. In this carboxysome, the bicarbonate is transformed into CO2 by carbonic anhydrases, and this raises the CO2 concentrations near the Rubisco enzyme, which has a relatively low affinity for carbon, for CO2. So this is a way to get high CO2 concentrations near the Rubisco enzyme for optimal photosynthesis. Now what we did is we sequenced the Microcystis genome for 13 Microcystis strains and looked at to what extent do they differ in the different components of this carbon concentrating mechanism. And we found, for instance, if you look at these transporters, there are these different uptake systems which are indicated by the colors here, and also here by the colors. We have 5 different carbon uptake systems, 3 for bicarbonate, the BCT1, SBTA, and big A, and 2 for CO2. And of these 5 systems, 3 were present in all the Microcystis strains that we sequenced. Then we also developed PCR techniques to investigate more Microcystis strains specifically for these uptake systems. And we found that also in these other strains, these 3 uptake systems were always present. However, these 2 uptake systems are not always present. They are variable. Some species lack both of them. Some species have them all. Some species have only one of these 2, either SBTA or big A. And these 2 systems, SBTA and big A, also differ in their properties. So SBTA is a system with a high affinity for bicarbonate but a low flux rate. And big A is the reverse. It has a low affinity for bicarbonate but a high flux rate. Now what happens in many of these Microcystis dominated blooms is that if you take a sample and you analyze it, you find out that it's not a monoculture, but it consists of many different strains with different properties. For instance, it's often a mixture of toxic and non-toxic strains. You see an example from a lake study. During the summer season, you see how the strain composition of a bloom changes over the summer season. And the strains we now know can also differ in the carbon uptake systems. So this is for different Microcystis strains, we show the different uptake systems. For instance, this strain has only the big A. These strains have only the SBTA. Some of them have a big A, but the big A is interrupted and is not functional. And these strains have a functional big A and a functional SBTA. Now what we did is we also performed growth experiments to investigate what is the consequence of the presence of these uptake systems for the growth rates of the organisms. We did this for 3 strains. This strain here, PCC 7806, is only big A. And big A was the enzyme with a low affinity for bicarbonate with a high flux rate. And what you see here is that at low PCO2 levels, this strain performs poorly. It has a low growth rate in normal medium, and even if you add buffer, which raises the bicarbonate concentration, its growth rate is lower than in compared with higher PCO2 levels. Strains that have both big A and SBTA perform quite well at all the PCO2 levels, and strains with only SBTA, so high affinity but low flux, they perform well at the low PCO2 levels, but they perform less well at the high PCO2 levels. So you see really also phenotypic implications of these different uptake systems. The next step was to do competition experiments, also in a laboratory chemostats. So here you see some examples. At the low PCO2 and at the high PCO2 level, we took a strain with only SBTA and compared it with a strain with both big A and SBTA. And then at low PCO2, the strain with only SBTA1. So the selection for SBTA, which has a high affinity but a low flux. So that's logical, that's what you need at a low PCO2. And here at high PCO2 levels, we see that the strain that had both big A and SBTA was winning, so there's selection for big A for low affinity but a high flux rate. So the implication for this is that differences in environmental CO2 levels will select for different strains, for strains with different carbon uptake systems. In this particular case, I haven't shown that here, but the strains also differ in their toxicity. So you get not only changes in the strain composition, but you also get changes in the toxicity of the blooms.
And they must get worse.
In this example, no, they get better. The toxicity declined. But the thing is that the toxin genes and the carbon uptake genes are not coupled. So it's just, if we had chosen other strains here, then we would have gotten other results on the toxicity. But you will get changes in strain composition, which might give rise to changes in toxicity of the blooms. Yeah. So the take-home message of this story is, algal blooms can deplete the dissolved CO2 concentration and increase the pH. They are predicted to do this over a wide range of atmospheric pCO2 levels, but at higher pCO2 levels, the increase in pH will be less, and the depletion of dissolved CO2 by algal blooms will also be less intense. What will intensify is the intensity of the algal blooms, especially in low alkaline eutrophic and hydrotrophic lakes. And what we may also expect is that rising pCO2 will cause changes in the strain composition. All right.
And are the strains which are non-producing toxins, are they susceptible to the toxins by the others? Because this would also be an explanation for the toxins that they
Yeah,
each other.
but that doesn't happen now. It has been investigated, but there are allelopathic effects. And there are some early publications which have also suggested this, but there's now not a strong evidence for it. No.
And what proportion of them, are they all toxic?
Are they all toxic?
Okay, what proportion of?
Oh, we can actually have a look. So if you look a bit more in detail, here these are lake data. So this is based on old-fashioned DGGE, looking at relative band intensities. It was in 2007. But you see here we have some strains are non-toxic, some are toxic. And if you look at the lake, you always find some toxic and some non-toxic strains. But some cases the blooms are dominated for 90% by the toxic ones. Some cases they're dominated for 90% by the non-toxic ones. And really this change in the relative abundance of the toxic versus non-toxic strains determines the toxicity, strongly determines the toxicity of a bloom. And we think that's also why, for instance, in Lake Volkerak, we have every year cyanobacterial blooms, but only in the year 2002 and 2003 it caused mass mortalities of birds. But also those 2 years coincide with very high levels of, very high relative abundance of the toxic strains.
And do the non-toxic ones have a higher growth rate? Because they need not invest into toxicity?
Marginally. So the costs of the toxin production are not that high. There is a very high cost of the gas vesicle production, but the toxin production it's apparently rather cheap for them. Yeah.
So how many, so these are genes, is that really, so if you look at the strain level, how much variation do you think there is? Has anyone gone out and really sequenced?
Yeah, sequencing has also been done. And in this old-fashioned DGE, we could find like up to 10 different strains in the lake. With sequencing, you could find 30, 40 different strains.
Okay, but you can find like individual clones which are at appreciable frequency.
Yeah.
But there's like, well, 30. Okay.
So it's really a diverse mix. These blooms are not just monocultures, it's a diverse mix of different strains. That changes over the season.
So you can't kill it with one phage.
Yeah, depends on the specificity of the phage. Yeah.
You find the superphage.
A superphage, yeah.
Superfibers in that case.
And we've also used several other treatments for lakes, for instance, hydrogen peroxide treatments. That also works against cyanobacteria because they're much more sensitive to hydrogen peroxide than the eukaryotic phytoplankton. So that there are other approaches as well. But it's all expensive.
Especially with a big lake like.
Yeah, Lake Taihu, it's incredible. I don't know how one could solve that with 2,000 square kilometers.
Yeah. I have a question for your carbonate chemistry. Can you go back to the slides show the different carbonate species? Yes. How do you analyze CO2 out of TIC?
Let me see, because my co-author, or my first author, Yolanda, knows more about this than I do. What I know is he measures total DIC,
Yes.
then he titrates. Because I know part of it is calculated, but I do not know whether she calculates the bicarbonate or the carbonate or the
I think.
so you measure pH, you measure total DIC, you measure alkalinity, and I think the rest is calculated from the chemical equilibria.
Okay, so you assume there is a chemical equilibrium between CO2 and bicarbonate and carbonate.
Yes, but not an equilibrium with the atmosphere.
Not an equilibrium with
Yeah.
atmosphere. Okay, so the dashed line here, CO2x, represent atmosphere CO2?
Yeah, that's an equilibrium with atmospheric CO2.
Okay,
So just at 380 ppm or something you would expect.
okay.
And then depending on the temperature, because the dissociation constants are
Okay.
temperature dependent, you get a bit of ups and
Okay.
downs.
All right.
Yeah.
Then can you move on to the slide shows the laboratory reactor experiment with the CO2? Okay, here.
Yeah.
So these CO2s are calculated. The CO2, bicarbonate CO3 is calculated from DIC and the pH. Is that
Yes.
correct? Okay. It is possible that, well, it's not possible. It is a fact that these species may not be at equilibrium.
Yeah, but if you look at the rate constants, like the development of the phytoplankton blooms are on a much slower time scale. We're talking about a time scale of days.
Okay.
Whereas the equilibration of between CO2, bicarbonate, and carbonate is in a matter of an hour or so.
So do you have any idea? Because, I mean, we see a lot of where the remineralization rate goes with the increasing temperature.
Would
starts
really go up.
goes really
Yeah.
quickly, right? And so then the algae even do better. Is that, have you done anything like to look at that? Do you have any idea in those lakes, like what the remineralization rates are?
No, we have a project proposal on this because we are very curious to know like what happens with the remineralization of the organic carbon and to what extent does this feed the phytoplankton in the lakes.
We'll actually see DOC go down during the bloom.
Okay.
Actually.
Okay, so it's really used rapidly.
It's really rapid, yeah.
Wow. Yeah. No, I think that's very interesting. So
Yeah.
we also.
There's also things Stat has a paper where they saw that in the Arctic.
Okay.
So this would seem a very, very tasty dinner for us.
Yes.
So why did, so it can happen?
Yes, it's known that some blooms collapse because of a virus infection. Yeah, you can also mimic it in the laboratory. Yeah.
Okay, why do most blooms not collapse? According to your view of the world, they should. I don't know.
Well, they have to find the right virus, right? I mean, it's putting the right things together to get it to happen.
Yeah, it's a fake answer, but I don't know the answer either.
Well, these guys are special, right? So they're like us. They're not bad.
Yeah.
They're not homogeneous at all.
Yeah, I know, are they not? Well,
But for instance, for Lake yeah, no, it's
you
out
saying
of
that, you're
bloom.
saying if you look at one of these big lakes, you're saying that there isn't there'll be
They're not
thousands of
they're not isotopically the same. I mean, they're not isogenic, sorry.
there'll be
They're not
thousands and thousands in a big lake.
different types, yeah. I mean, not that you see at this level, because they're the same species in quote unquote, but there's a whole
But there are lots of different strains. So we see, for instance, where people think like, okay, the lake looks the same every year. We have again every year the same microcystis bloom, but actually the strain from one year to another
It's
can be
massive.
very different.
And that is driven usually by the viruses or the predators in that case. Because you'll see that it'll be like the receptors for the viruses or the, I mean, there's all, or the immune systems to the viruses and stuff of that nature. We don't know in microcystis, I don't think there's hardly any, there's only just like trying to kill them, right? But,
Yes,
yeah.
with all kinds of approaches, and a few people working with viruses.
So E. Huxley has, like there they know really what's going on in a big algae or an algae, a semi-big algae.
Yeah.
Where does it come from? So during the winter there's no microcystis and then it wakes up again. Was it in the lake the whole time?
It sleeps on the sediment.
Right.
Now what happens is they produce gas vesicles,
Yeah.
which give them buoyancy, and they also with photosynthesis they produce carbohydrates, which give them ballast.
Yeah.
And when the temperatures get colder, they produce less gas vesicles and more ballast. There's less respiration at lower temperatures, so they sink to the bottom, to the lake sediment. And for instance, Lake Volkerak, there's a pile like this on the sediment of microcystis. We have sampled it.
Then why do you get the same strain every year? Because you would have thought that.
Yeah, but also the sediment, we've also looked at the sediment, it's a mixture of strains.
Yeah. Okay.
And we think that part of it, we have done some, it's not in these papers, but we have written some other papers on like the benthic pelagic coupling of the ones on the sediment and the ones in the water column. And based on that, we estimated that each spring about half of the population comes from the sediment, because sediment resuspension occurs, but not that often. And half of it are the survivors that stayed in the pelagic, in the lake water. Yeah.
Okay.
Maybe it's related to your results that there is divergence,
No, I just
the
wondered,
unpredictability in the
Is it just
composition.
random
Yeah,
which
yeah,
one
yeah.
gets amplified in the spring?
Might be. Might also be some
Yeah,
pressures
some-
with
Some
viruses.
nonlinear feedbacks
Yeah.
which can lead to multi-stability.
Interesting questions.
I mean, also during your talk, I thought about, for example, grass habitats or plant habitats. So there are different composition of plants, and on one lawn there are some flowers predominant, and on other lawns other flowers, and it's maybe not predictable because there's some feedback off because the
Once
species
established,
who are there, they
yeah.
stabilize
Yeah.
themselves and
Yeah.
exclude
Yeah.
others.
Yeah, yeah,
It would be nice to know how.
Yeah.
yeah.
That's-
How and when.
Yeah.
Yeah. Okay, guys. Thanks.
Thanks.